Good evening, ladies and gentlemen. For those who don't know me, I'm Professor Paul Lynch, and as Chair of the University Professorial Council, I'd like to welcome you to the latest in the series of inaugural lectures here at the University of Derby. This time we've got Jose Prados, who is the head of psychology, the School of Psychology here at the University of Derby. I'd now like to introduce Dr. Denise Baker, who is the PVC Dean Interim for the College of Health, Psychology and Social Care, to introduce Jose.
Thank you, Paul. Welcome, everybody. It's my pleasure to introduce Jose to the audience. Jose was born in 1967, the third of four Prados children in Barcelona in Spain. He had an interesting childhood that involved the ever presence of his maternal grandmother and 60-odd cousins. Being asthmatic, he benefited from being away from the big city, so spent some years in a Catholic boarding school in the Pyrenees near Andorra. Curiously enough, his parents were members of the then illegal Communist Party, so diversity and contradictions were at the core of his education. In the mid-1980s, he started his degree in psychology at the University of Barcelona. On the first day of the autumn term as a fresher, one of his lecturers, Dr. Victoria Chamizo, introduced her module and told students that they would be assessed on the contents of a particular book, Animal Learning and Motivation by Roger Tarpey. She also asked for volunteers to work in the Animal Psychology Lab, and Jose instantly stepped forward, and this started his lifelong collaboration with members of the team. During the first year, he rote-learned, and that's not a recommendation, it's just a statement, he rote-learned to the contents of the Tarpey book and scored a ridiculously high 98.5% in animal learning. And a true oddity, somehow he wasn't as brilliant in other areas.
That happens.
That was the beginning of a long and colourful disciplineship to Dr. Victoria Chamizo, who eventually became Jose's doctoral supervisor. Chamizo, one of the leading experts in spatial learning, had a very close collaboration with Professor Nick McIntosh, the head of the Department of Experimental Psychology at Cambridge. Still as an undergraduate, Jose developed with Professor Chamizo and Professor McIntosh the Morris Pool facility in Barcelona. He later used this facility for his PhD project on pre-exposure effect on a navigation task with rats. His interests were still diverse, and he was simultaneously training as a family therapist. During his PhD, Jose benefited from long annual stays of several months in different UK universities, where he was lucky enough to collaborate with John Pearce in Cardiff and Geoffrey Hall in York, working on the associative analysis of spatial learning and perceptual learning. In 2000, Jose was among the first students in Barcelona and possibly in Spain submitting a doctoral thesis as a compilation of published papers. Following the award of his PhD, Jose held fixed-term lectureships in his home department. In this period, he established himself as an independent researcher funded by the Spanish Ministry of Science, but was growing increasingly dissatisfied with the lack of prospects in Spain, where even prominent researchers were only offered short-term contracts at the time. In 2004, Jose, his wife Nuria and baby Simon decided to make England their home. Jose was offered a lectureship at the University of Leicester, where he spent 17 fantastic years teaching and researching. He held a diverse range of roles, including Head of School of Psychology from 2017 to 2021. The Prados family welcomed two more Leicester-born children, Martin and Leon. In 2021, attracted by the immense talent and reputation of the psychologists here at Derby, he seized the opportunity to become the head of a newly established school of psychology, and a new adventure has begun. Ladies and gentlemen, I'm very pleased to introduce to you Professor Jose Prados.
Wow. And in the meantime, I can take the opportunity to respect convention and thank everybody in my life, past, present, and future, for the impact they are having in my work and in my personal life. I would say that both in the private business and in academia, our achievements, but mine in particular, are the product of a collective effort. So I'm really thankful to you all, and I'm really thankful for you to come today to this place. And now it's working. So now, into business. My title is The Evolution of Learning Towards a Philogenetic Epistemology. I like this title because most of people don't have a clue about what it is about. And that includes myself. I guess that the subtitle is redundant. It's actually meaning the same as the title. In a nutshell, I would say that our interest is to explore how learning emerged in evolution at some point, how it evolved, became gradually more and more complex, and how is it distributed in the phylogenetic tree. That would be my talk. Now you can go to sleep if you wish, and save the next 45, 50 minutes. I'd like to do that in 45, 50 minutes. Simon, can you set an alarm in 25 minutes and give me a warning, please? 25 minutes. And then we will see whether I keep pace or I accelerate. So this is it. Evolution. This is about evolution of cognition, and my focus today is going to be, one of the focus would be on the emergence of consciousness. So we have been interested in consciousness forever, but nowadays it has stopped being merely or just a philosophical issue. It is now the subject of empirical investigation. From an evolutionary perspective, we consider cognition and consciousness as products of biological evolution. And the main question that we want to answer, that we want to address, is who, and by who I mean what animal species actually have a conscience, have the capacity to experience conscious states. It might be just the humans, and perhaps not all the humans. It could be humans and great apes. It could include some birds, some fish. The question is whether animals, relatively simple in their brain architecture, are capable of experiencing conscious states. The definition of consciousness is, of course, of relevance, because if you define consciousness in a very constrained manner, you are reducing the possibility of finding evidence only in humans. If you are too liberal, then you become a pan-consciousness theorist, and you end up claiming that inanimate objects and the universe also have the conscience. And this is a big stretch. I suppose that we will stay a little bit in the middle. So the way in which we are going to address the presence of consciousness throughout the animal kingdom and beyond is by the use of Pavlovian tasks. I've been introduced more or less as a learning theorist. The only thing that I know how to do is Pavlovian conditioning, and my plan is to use Pavlovian conditioning tasks to assess the cognitive abilities of different animals and see whether they actually meet the criteria to be considered conscious. This is one. Okay, so what is consciousness? This is the first question that we have to address. Consciousness is the capacity to have subjective experience. The question is that we can distinguish different forms of conscience. So some beings can be sentient, can experience sentience, and this means that they can experience feelings, but they are conscient only at a very low level. It's a very elemental form of consciousness. You can have feelings, but you might not be able to consciously reflect on your feelings, and of course, you might not be able to be aware of the feelings of other people or other individuals. So sentience is a first step, I suppose, in evolution towards consciousness. Being conscious is a different thing, it's more sophisticated, gives you the capacity to have subjective experience, some agency. You have a sense of your individuality. You can distinguish yourself from the background, from the world, and you have a subjective point of view of yourself and the world. If you are conscious in that way, you might be able to show, to display goal-directed action, so to behave with purpose. And if you are conscious in that way, you might be able to guess, to be aware of the feelings, the knowledge, and the intentions of other individuals. This is the theory of mind. So if you are capable of recognizing that others have feelings and knowledge and intentions, then you are likely to behave in a compassionate manner and behave with humanity. Okay, so consciousness is something more complex than mere sentience and gives up, defines at the end of the day what being human is about. But we are sharing that, this is my hypothesis, with other beings. The question is, consciousness must have evolved, must be the product of biological evolution, and we could identify a line that separates those that can experience conscious states from those that cannot. Or even we can discriminate between those that can have feelings from those that cannot have feeling, depending on the biological machinery and the cognition that they have evolved. What is the line? Okay, to help with establishing where is the line, to discriminate who qualifies for conscience or not, we can use the work of Simona Ginsburg and Eva Jablonka. They've been working on that issue for the last 20 years, 25 years, perhaps all their lives. They came up in 2018 with this book, which is a big one, and they make a number of proposals which are really useful to organize the field of invertebrate learning. In this book, what they suggest is that unlimited associative learning, something that they refer to as unlimited associative learning, can be an evolutionary transition marker for conscience. That means that if you assess the behavior of a particular animal species and they are capable of showing unlimited associative learning, then these animal species would qualify for having conscience, to experience conscious states. So the line could be here, separating humans from the rest of the organisms, and this would be the new hypothesis. We know that we have conscience. And we can identify different sorts of learning or different types of learning: very simple habituation and sensitization, limited associative learning, and unlimited associative learning. By assessing the cognitive skills of different animal species, we can draw the line here or here. Perhaps the vertebrates and the octopus, which has a special status, are capable of consciousness. Perhaps it is the animals with a brain, with a central nervous system, and perhaps it can go beyond that, but I would be reluctant to go further. So beyond this line, there would be animals with a radial, non-centralized nervous system without a brain, and there would be, I don't know, plants, fungi, protista, cells, etc. This is what they are proposing. So before we continue, I already said that my cunning plan, is to use Pavlovian conditioning for everything, simply because this is what I know how to do. A little bit of instrumental conditioning, but today I'm focusing on Pavlovian conditioning. Is there anyone in the room which is unfamiliar with Pavlov's dog? Leon? You are unfamiliar with Pavlov's dog? Okay, for the sake of Leon, I will explain the basic paradigm. Remember that Pavlovian conditioning is a form of learning which allows individuals, animals, for example, to learn about the causal structure of the environment. They can identify signals and relevant outcomes in their environment. So this is great because then they can change their behavior during their lifespan, and they become more adaptive. They can adapt to the changing conditions of the environment, and they have more chances for survival and reproduction. So Pavlovian conditioning is a very powerful tool to have for an individual, an animal or a human. And it allows, well, some people even claim that associative learning, Pavlovian conditioning, instrumental conditioning, has been the main driver of evolution. That at some point in the Cambrian period, for example, the emergence of associative learning was responsible for the huge diversification in the animal kingdom that we can see until today. So the paradigm that Ivan Pavlov came up with was, playing with dogs. And basically with dogs, we know that they are very interested in food, and you know that. Yep. And when they are presented with food, they would naturally salivate. They will automatically respond to this biologically relevant stimulus. When they are presented with a bell, the bell that never sounded in Pavlov's lab, in the presence of a bell, they can react. This is a relatively neutral stimulus that would provoke an orienting response, but the animal will not drool in the presence of the bell. So the Pavlovian paradigm consists of pairing the neutral stimulus with the biologically relevant stimulus, the unconditioned stimulus, and after a few pairings, if you present the initially neutral stimulus, the bell, then the animal will drool, will salivate. The notion is that during conditioning, the animal learns an association between the neutral stimulus and the unconditioned stimulus, the food, and at the time of test, when you are presenting the bell, the animal activates a mental representation of the food, and this is what provokes the conditioned response of drooling. So far, so good? Have you learned something today, Leon? Good. So this is what we are going to use in our work. We now need to differentiate, distinguish between limited and unlimited associative learning. This is of importance because according to Ginsburg and Jablonka, unlimited associative learning would be a marker, an evolutionary transition marker for consciousness. And how do we do that? Well, I don't really know. I keep reading their papers and their book, and I always struggle with the way in which they are defining this. So I'm going to do it in my own way. And one way of distinguishing between limited and unlimited associative learning is by reference to contiguity. So in the Pavlovian paradigm, for example, one of the conditions that can be observed to ensure that there is learning is that the conditioned stimulus and the unconditioned stimulus are presented close together in time. There needs to be some contiguity between them. The closer they are in time, the faster the animal would learn an association between them. And some people have referred to this kind of learning as Hebbian learning. I don't know whether this is entirely fair, but at the time I was writing a paper in 2012 or so, and I distinguished between simple and complex Pavlovian conditioning, which was brilliant. But the reviewers didn't like it, so they suggested using Hebbian, based on the notion that when the light and the shock, for example, are presented, some neurons in the brain will be activated, and according to the quotation, neurons that fire together, wire together. I don't think that Hebb actually said that, but the idea is that if there is co-activation of the neural centers responsible for the processing of the light and the shock, then an association will establish between them automatically. So contiguity certainly works. If you present two things close in time, they will become associated, but this is not always true. And this is the second time that we are addressing contiguity today here. Contiguity is often not a sufficient condition for learning. In many cases, the learning in a Pavlovian task is modulated by other cognitive devices. For example, selective attention can be involved, filtering the information, or the animals can integrate information from different learning episodes which challenge the principle of contiguity. So let me show a couple of examples just to demonstrate that contiguity is not always necessary to obtain good Pavlovian conditioning. So the first one is latent inhibition. This was discovered by Robert Lubow back in the 50s, and this is based on an experiment published in 1965 that was done with goats. In that case, two groups of animals were trained in the standard Pavlovian conditioning task. A light was the signal for a shock and conditioned stimulus, which was delivered to the rear leg of the animal, and the shock resulted in the flexion of the leg. So with sufficient training, the light would acquire the properties of an effective conditioned stimulus, would elicit this leg flexion response. So the two groups were trained in these tasks where there is contiguity between the light and the shock. Consistently, the light was always followed by the presentation of the shock, but in one of the groups, the animals were made familiar with the light before the onset of the training in the conditioning task. So there were like 20 or 40 presentations of the light before the actual training. And pre-exposure to the to-be conditioned stimulus resulted in a deficit in learning. Here, what we can see, or here, if you prefer, what we can see is that I might be out of camera. Let me relocate. This is the performance of the group given pre-exposure that was familiar with the light, and this is the control group. This is the normal, the baseline acquisition of a conditioned response, and this difference, this deficit, is what latent inhibition is about. So, in spite of the contiguity between the light and the shock, the association was hindered. Why does this happen? Well, something happened during the pre-exposure. The animal learned something. Apparently, the attention paid to the light went down, and at the time of conditioning, the animal was not paying attention to the light, which loses then capacity to enter into new associations. So to account for latent inhibition, contiguity is not enough. We need to refer to selective attention processes. And this would be one of the characteristics of the unlimited associative learning. So far, so good? Yeah, really? Sure? Thank you. This is a nice reward. Another example, which is even better than this, is sensory preconditioning. Pay attention, Liam. Sensory preconditioning is the twin brother or sister of second-order conditioning. And this is an experiment in 3 phases. And again, what we are doing is simply training 2 groups of animals in a standard Pavlovian task, where the light becomes the signal for a shock. This experiment was done by Brogden in the late 30s with dogs, and by Risley and Rescorla in the early 70s with rats. Before training in the conditioning task, one of the groups had some experience with the to-be conditioned stimulus and a third stimulus, in that case, a tone. So in the case of Brogden, a tone and a light were presented together simultaneously as a compound. In the case of Risley and Rescorla, the tone preceded the presentation of the light over a number of trials. A control group, and there were different control groups in these experiments, a control group were given exposure to the tone and the light, but at random. There was no way in which the animals could establish a relationship between these stimuli because there was no relationship. And the final test was done with the tone alone. What they wanted to know was whether this experience in phase 1 would affect how the animals behave in front of this tone stimulus. So what can happen in this kind of experiment? In the first phase, the tone might become a signal for the light. In the second phase, in both groups, the light could become a signal of the unconditioned stimulus, would become an effective conditioned stimulus, and would result in conditioned responses, fear conditioned responses. It is important to note that in this design, come on, in this design, there is no contiguity between the tone and the shock. The tone and the shock have never been presented together. And furthermore, when the tone was experienced with the light was not yet an effective conditioned stimulus. So there is no way the tone could be linked with the fear response I told you in the first phase of the experiment. In spite of all these limitations and the lack of contiguity, when they tested the animals in the group given pairings of tone and light and shock showed an effective fear conditioned response. So the tone was an effective conditioned stimulus in spite of the fact that it had never been paired with the unconditioned stimulus. There was no contiguity at all. The only way in which we can account for that result is by assuming that the animal established different associations, acquired different information in the two learning episodes, learned first to link the tone with the light, and then associated the light with the unconditioned stimulus. And this associative chain would allow the tone to be an effective conditioned stimulus. So to account for the sensory preconditioned effect, we need to assume that animals are able to integrate information from independent learning episodes. The animals learn things at different times that then can be linked together, and this changes the way in which they behave in front, in that case, of the tone. Impressive, isn't it? Okay, so far so good. Time? 4 minutes, 21 so far. Okay. So, this is the most difficult one, I think. Ginsburg and Jablonka came up with 5 key features that define unlimited associative learning. To be honest with you, of the 5, I have selected 4 because the 5th I couldn't understand. And I have replaced that by one by myself, which is in green. But basically what they are saying is that to be considered unlimited associative learning, the animals must be able to use selective attention, integrate information from different learning episodes, show flexibility, so one conditioned stimulus can change value if the conditions in the environment have changed. So something can be indicative of something appetitive one moment, and then this can change if the conditions in the surroundings have changed as well. It needs to be complex. The hierarchical structure or the association structure behind learning can be hierarchical in such a way that one stimulus can become, for example, a retrieval cue for the association between other two stimuli. So the context, for example, can activate the association between two stimuli. And finally, the animals must be able to distinguish the compound from elements. So A and B together are not the same as A and B presented by their own. This defines unlimited associative learning and requires some level of cognitive sophistication. Okay? So with this in mind, then I perpetrated that. This is my contribution. This is a very nice slide. I think that the color palette is nice. And, well, this is as far as I go. And I've captured the 5 conditions for unlimited associative learning here. And I've identified a number of basic learning phenomena, Pavlovian phenomena, that would require the usage of these skills. So this goes from very simple learning, non-associative, sensitization, short-term habituation, to limited associative learning, to unlimited associative learning, and then this is where my wisdom reflects. Superior cognition, whatever this means. Superior cognition would be, I don't know, episodic memory, the capacity to learn the past, plan for the future, and theory of mind, be able to read the minds of others, be aware of the intentions and feelings of others, tool usage, I don't know, tool usage should be here, the capacity to interact with the environment by using things which are external to your own body. All this might be superior cognition that depends on mechanisms that I have labeled as complex systemic brain activity, meaning I don't know. This is for my colleagues to feel. I suppose that the cognitive neuroscientists in the room might have some ideas. So basically what we want to do is to assess which animals qualify for unlimited associative learning. And I would say that humans and vertebrates in general are typically assumed to be able to show unlimited associative learning. We have seen that dogs and rats are able of integrating information from different sources and that kind of things. We know that organisms from other kingdoms, protista, or plants or fungi, are able to learn. They learn from experience. Their behavior can be changed as a consequence of experience, but typically we assume that they would show very low-level learning. And then somewhere in the middle, we will have the other animal phyla. And by this I refer to, in general, invertebrates. So there is the molluscs, the arthropods, the nematodes, the earthworms, rotifera, etc., etc. There are many of them. So these other animal phyla, the invertebrates, might sit here, according to some, or according to others, they might be as competent as the vertebrates and would show unlimited associative learning. I suppose that they sit in the center, in the middle, if I can get this working. And what we need to do, our task is to assess whether different species representing the animal phyla actually meet the criteria for unlimited associative learning. How am I doing? Is this okay? Poof, it's warm in here. This is the phylogenetic tree in a circular fashion. I like it because this shows us where we are, which is here. This is the Homo sapiens. Sits among thousands of species in the animal kingdom. Sits among millions of species in general. So this is just to humble ourselves. We are just one species among many, many millions. The plan for today is to review some evidence about the learning abilities of different organisms. I'll start very basic with a protist. Protist used to be, I don't know, because the biologists keep changing the names for these things all the time. They are single cells, complex cells in many cases, but just one single cell. I'll then review two experiments with plants, which are of some merit, and then I will jump from the plants to animals with a brain, animals with a centralized nervous system. I'm skipping the animals with a decentralized nervous system without a brain because there is really not much evidence about them. They have not been really studied. And starting with the protists, starting with the single cells. There is a long tradition that goes back to the early 20th century, suggesting that single cells, protists, like the Paramecium caudatum, can learn, and many important, very important people at the time even claimed that these single cells behaved with purpose and have conscience. I think that this is a very, very long stretch. But they certainly have some learning capacities. So this is an experiment that was published by Hennessy, Rucker, and McDermott in 1979. And I favor this one because this was published in Animal Learning and Behavior. And for some reason, I tend to trust what is published in that particular journal. So this is a capillary tube, and the authors of the study simply capture one single cell in this capillary tube with the medium, and they could pass an electric shock through the medium. The paradigm that they used was a Pavlovian conditioning paradigm, and they simply presented a conditioned stimulus, or to be conditioned stimulus. It was a vibration, that was produced by a little speaker close to the capillary tube. This is done, by the way, under the microscope. And following the presentation of the to-be conditioned stimulus, they presented a shock, and the presentation of the shock produced an unconditioned response of axial spinning. So they repeated that a number of times. Oops, what is that? And by pairing the vibration, the conditioned stimulus, with the shock a number of times, at some point they discovered, or they found, that the vibration by itself produced the axial spinning. They ran several experiments. I'm going to tell you about two of them. This is not entirely okay. This is the first experiment where they trained two groups of cells, and one of them was given the standard Pavlovian training. The vibration was followed by the shock in every trial, and the second group was given random presentations of the vibration and the shock. And what they found was that those cells that were given per presentations of the vibration and the shock actually developed a good level of conditioned response. This is the probability of showing the axial spinning in the presence of the vibration alone. And you can see that there is some level of response in the control condition, but is not as high as the one in the group given the pairings between the CS and the US. So this difference here seems to be good evidence for Pavlovian conditioning in single cells. Not happy with that, in a subsequent phase of the experiment, they omitted the unconditioned stimulus, the shock, and they saw that after a number of trials, the conditioned response extinguished. So this is a basic characteristic of any Pavlovian conditioned response. Looks like single cells are capable of learning the relationship between two events in a Pavlovian preparation. In the second experiment, they presented again in one of the groups on the day one of the experiment the vibration followed by the shock, and then they waited 24 hours before continuing with the training. In the second day, they added two more groups that were given the vibration with the shock or vibration and shock at random. What they wanted to know was whether a single cell, a single complex cell, would be able to retain the information acquired in day one, and this would facilitate the acquisition in the second day of the experiment. So this is a memory experiment. And what they found is that the group that was pre-trained in day one actually showed a great advantage. So the cells, not the animals, not the plants, the single cells actually retain the information for 24 hours. And this seems to demonstrate that not only can single cells learn a Pavlovian association, they can retain the information long term. This is an instance of long-term memory. So how does this happen? Well, we have to take the bullet. I was very skeptical about that kind of things, but there is a number of experiments, classic experiments by Beatrice Gelber in the 50s, and some more recent experiments by a guy whose name is Harvard Armus that have been replicating the classical studies, and there is evidence piling suggesting that individual cells can actually show Pavlovian conditioning. So this is happening. They can show long-term learning and memory. The question is how, and by how we need to look again at the work of Ginsburg and Jablonka, and they have suggested that learning requires an epigenetic process. And this is common to neural animals and single cells. So what we understand nowadays is that when they are exposed to the learning experience, to the events which are part of the Pavlovian paradigm, this switches on one gene, which then triggers the transcription process, And the result of that is a messenger RNA which is transported out of the nucleus, and the ribosome reads the code and produces a new protein. So the synthesis of this protein is the product of the learning episode. And this new protein actually changes the structure of the cell, which will now react to the conditioned stimulus in a different way than they reacted initially. So they would hopefully react in a more adaptive way. This is common, I said, to the single cells and the neural animals. Protein synthesis is necessary for long-term encoding of information. If you treat the animals, rats for example, with an inhibitor of the protein synthesis, they fail to remember the information acquired in a learning episode. So, this suggests that the emergence of learning was very early in the day of evolution. It's a very old mechanism that was already present in individual cells, and this might have been maintained throughout evolution and is now in use in specialized tissues that process information. That would be the nervous system of the animals. So far, so good, Leon? Good. He's the younger and the less qualified in the room, I guess, so it's a good test. So let's jump from single cells to a complex organism, plants. Do plants learn? Remember plants? They are at home doing that. He says no, they don't learn. I would agree with you, because plants are really difficult to deal with. I mean, they don't do anything except a few species which actually react immediately to a stimulation. So Mimosa pudica is one of those species that present a very clear response to mechanical or electrical or chemical stimulation. When you straw one of the pinna of the Mimosa pudica, these little branches referred to as a pinna, they fold immediately. Okay? And this immediate response, this behavior, has been used to assess whether plants can learn or not. I was a skeptic, as you can imagine. I mean, plants learning, this is beyond boundaries, really. But in 2014, Monica Gagliano, who is actually quite famous, came up with one paper in which she demonstrated long-term habituation in plants. It was beautifully written, it was very convincing, very persuasive, and I was still not buying it. And simultaneously, in 2014, Sabrina Amador Vargas came up with another paper with Mimosa pudica, again demonstrating habituation learning. The two studies differed a lot. So Monica Gagliano was stimulating the whole plant. Basically, what they did was to drop the plant pot from a height of 15 centimeters, and this is anything but subtle. It was like plok, and then all the pinna closed. And Sabrina Amador Vargas was more subtle, and she was stimulating just one leaf of the pinna. They dropped a grain of rice on one leaf, and then they measure the folding of the individual leaf. And in some cases, I lack the expertise to deal with plants, to be honest with you. So I was struggling to believe that this actually happened. So I decided to do some experiments with plants myself, and rather than doing the brutal stimulation of the whole plant or the subtle stimulation of one leaf, we decided to stimulate one pinna and see how the pinna folded. So to do experiments with plants, you need to be well prepared. You need a very rigorous protocol, and in my case, the protocol took like 7 steps. The first thing is that you cannot do that alone. You need to do that with a research assistant, which needs to be motivated and needs to have the right expertise. And I was lucky enough to get this guy here, who helped me planting the seeds. And then we, over days and weeks, we nurture the seeds. We respected the conventions about temperature and moisture. The seeds germinated, they grew, and what else? We transplanted them, or whatever it is in English. We well, we engaged in mass production because I had the plan of running complex experiments with different groups and different independent variables. I was ambitious at this stage. And when the plants were sufficiently grown, then you could do two things. One was to do experiments, which is the boring one. The other one was to bring them to school for show and tell. Remember? I wonder what happened to this plant with 20 boys poking at them over one day. So this is what Leon did, and this is what I did. I brought the plants to the lab, and we wanted to measure the folding response and the reopening. How do you do that? Before stimulating the pinna, and we did that with a little brush, we simply strode the pinna and it folded. Before that, we measured the distance between two of the leaves. We went for the fourth row, for example, and this was giving us the maximum distance. So after a few seconds, what you get is the minimum distance once the pinna has folded. And if it is 1 millimeter, then you get a percentage of the maximum distance, which is in that case 8%. So 0% would be the total closure, 75% would be a weak closure, weak folding, 100% would be absence of folding. And then you can monitor the reopening. So after 10 minutes, if it is, for example, 6, you are already at 50%. And after about 20 minutes, we were expecting the plant to be back to normal, back to the 100%. Something I learned is that if you are not an expert in plants, then you cannot do big things. The simpler the experiment, the better. We did several things with different students. We tried to use electrodes, and it was a complete disaster. But when the manipulation was simple, really, really simple, we got some decent data. So Jack Hughes, some in the room might remember Jack Hughes. Do you? You do. We did this experiment in which we simply trained the 6 plants on an habituation task. So we simply stimulated the pinna 8 times within a session, and our expectation was that the first time they would show a strong folding response and a slow reopening, and then in the trial 6, 7, and 8 of the same session, they would habituate, and the folding would be weaker, and the reopening would be faster. But what we found was the opposite. Within a session, what happened was that the pinna closed the maximum closure possible, and then the reopening was slower and slower. So Jack was expecting to spend like 3 hours in the lab, but he spent the whole day, 6, 7, 8, 9 hours, I don't know, because the plants didn't reopen at the pace that we expected. So after the first day, having spent like 8 hours in the lab, I managed to persuade Jack to return on day 2 and repeat the operation. After the second day, he thought it was a complete disaster because we were looking for habituation and we got sensitization. And I couldn't convince him to come back the third day. It was a Wednesday. He said he had an excuse. I have a deadline for an essay. So I said, okay, when is the deadline? Thursday. Come back on Friday. A bit of negotiation. He came back on Friday. And then, after another long session on Friday, somehow, I'm fantastic, really. I persuaded him to come back on Monday and repeat this nightmare of experiment for the 4th time. And the reason to be so persuasive was that I had a cunning plan. I had realized something between the day 1 and the day 2. And it was that rather than looking at the evolution of the behavior within the sessions, you should look at the evolution of the behavior throughout the sessions, comparing throughout the sessions, and especially looking at the first daily trial. So this is what we observed the first time that we stimulated the pinna. There was a closure of about 12%, which is quite big, and then the reopening took this shape. It took like half an hour for the pinna to reopen in full and simply report in 20 minutes. Second day, we got this, which is a similar folding response, but faster reopening. And this faster reopening would be indicative of habituation. But not only habituation, it's long-term habituation. The experience of the training session 24 hours before was increasing the speed of reopening and on the first trial of the second session. Then in the third session, what we observed in the first trial was this, something similar. And this is good because it shows that in the second day we didn't get an artifact, so this was for real. And this is the fourth session. So what we got was a significant effect of reopening compared to the first day in days 2, 3, and 4, the first trial. And on top of that, we got on the fourth day a weaker folding response. And by itself, this would be just encouraging evidence. But taking together this and the papers from Amador Vargas and Monica Gagliano, I was convinced that plants actually learn. Plants show long-term habituation. How do they do that? I don't know. How? It's beyond my understanding. For the short term, habituation, ionic channels have been suggested, but for this to be retained over 24 hours, there must be a structural change in the plant that accounts for that. And I have no idea how this works. This is for the plant experts to decide. So I thought that I had some cool results with the long-term habituation, and then Monica Gagliano came up with this. This was published in scientific reports, and it's learning by association in plants. So she trained plants in a Pavlovian conditioning task, and she got actual Pavlovian conditioning, which is blow-minding. Don't you agree? So what did she do? Rapidly. So they exploited the fact that plants orient to the light. Light is very important to them. Light is the unconditioned stimulus. They found that if a plant, this is a pea plant, or a representation of a pea plant, is put in a Y maze and a blue light is presented in one of the arms, subsequently, when the light is off, the plant tends to orient towards the arm where the light was presented. And this is considered an unconditioned response. So they, in the proper experiment, what they did was to present together two events, the blue light and an air current, which was produced by a fan. And the rationale was that the air current would become a signal for the blue light. And they expected this effective conditioned stimulus to attract somehow the plant, even if it was presented in a place where the light had never been presented before. So in the test, what they did was to present the air current in the opposite arm, and the unconditioned response would be to go to the left, but the plant orientated towards the air current. So the air current was working as an effective conditioned stimulus, as a signal for the light. And this is an example of a conditioned response. And if this is cool, wait a second and see, because the last experiment was amazing. In that case, they presented the air current and the light in the opposite sides of the maze, in such a way that, if anything, the air current could become an inhibitor, a signal for the absence of light. And then they presented the air current in the arm where the blue light had been presented, and in spite of that, the plant decided to grow in the opposite direction to the air current. So the air current was actually a conditioned inhibitor, and what they demonstrated, according to that, was an inhibitory conditioned response. Amazing. I mean, this is beyond my expectations. So plants show associative causal learning, and I say, do they? I'm emotional now. No, I'm not. Do they? To be honest with you, I lack the expertise to properly assess these experiments. They are very persuasive, and I have to buy it. I know that some colleagues in plant learning labs, this exists, are interested in replicating, and I would like to wait until this is replicated independently by other researchers before concluding that causal learning can be observed in plants. And as for the how they do it, Your guess is as good as mine. Honestly, I have no idea. I have to finish in about 7 minutes. Okay, thank you. You're so generous. I'm jumping now from plants to animals, and these are animals with a proper brain, with a centralized nervous system. I'm starting at the bottom of the scale with planarians. Planarians are members of the phylum of Platyhelminthes, and this is the phylum which is farther away from vertebrates in the phylogenetic tree that still retain the same nervous system structure with a brain, and there are similarities between the nervous system of the planaria and the vertebrates, including mammals. So the neurons are quite similar, and they express the same neurotransmitters, and according to our research in the lab, with the same function. So the dopamine antagonist has the same effect in the planaria in a condition-place preference task as it has in rats. So planarians are suitable for learning studies, and the question was whether planarians would be able to show evidence for unlimited associative learning. So in that kind of experiments, I forgot to show you this, which is, what are we measuring with the planarians? Well, it's this, if it works. When you stimulate the planaria, mechanically or electrically, they show this shrinking response. So if you shock them or you touch them, they immediately shrink. And we can use that as an unconditioned stimulus. So in the experiments that we run with the planaria, we present it as the conditioned stimulus, light or a vibration, counterbalance. In principle, the light and the vibration were of lower intensity and didn't produce any apparent response. And the unconditioned stimulus was a shock. In this kind of experiments, in this one in particular, we train the animals in the Pavlovian task, this time with a compound stimulus. A and B, light and vibration, were presented together as a compound as signals for the shock. The animals shrink in the presence of the shock, and then during the test we presented one of the elements to see whether in the presence of B they show the conditioned response of shrinking. So far so good? One of the groups, however, had previous experience with the other element. So in the blocking group, the A element is pre-trained as a signal for the shock. And we know from the rodent literature and the human literature, the vertebrate literature, there are thousands of demonstrations of the blocking effect, that when you do that in the blocking group, pre-train A and train A and B together, B doesn't acquire the properties of an conditioned stimulus. They fail to learn about B. So B, which is here a redundant cue, A is a signal of the unconditioned stimulus. When you present A and B, A is already a signal of the plus, and B is redundant, and somehow animals filter out this B element, which doesn't become associated with the shock. The question was whether we could replicate this experiment in planaria, and the results simply show that in the first two phases of the experiment, all the animals in the control and the blocking condition acquired the conditioned response as expected, and when we tested B, we observed a deficit, the characteristic deficit of the blocking effect in the experimental group. Okay? So we replicated the blocking effect in planaria. And this is big, really. I mean, it took quite a lot of stamina to complete these experiments. This, by itself, challenges the principle of contiguity. B and the outcome are contiguous during the training. So there is something here that is more important than mere contiguity. And this extra thing could be the predictive value of A, which is carried from the first phase of the experiment to the second, or it could be selective attention. The animals filter out B, which doesn't have access to the learning mechanism, and there is no opportunity for B to become associated with the unconditioned stimulus. So flatworms seem to qualify for unconditional, I'm saying that wrong, unlimited associative learning. I'm highlighting that in yellow because I would like to be cautious, and before we claim that they are capable of unlimited associative learning and conscious states, I would like to see other phenomena to be demonstrated in the flatworm. This is annoying. Okay, the experiment number 5 is with snails. Just to show you, snails are one member of the molluscs, gastropods, are they? And the kind of experiments that have been done with snails involve snails being snails, and then snails eating. The key aspect is that when they are eating, this is a piece of carrot, the tentacles are lowered. And this is a natural reaction to which appears when they are feeding. So someone came with the idea that you could present, for example, an odor, the odor of a fruit, simultaneously with the feeding, and this would be equivalent to the A+ treatment. The presentation of a conditioned stimulus, the odor of the fruit, and the access to the food, which is the biologically relevant stimulus. During the eating, of course, the animals lower the tentacles. And then when you test the odor by itself, if the animals lower the tentacles, that can be considered a conditioned response. Okay? So using this procedure, my friend Nacho Loy, has run, I don't know how many experiments in Oviedo, and I want to highlight this one on sensory preconditioning. Remember, sensory preconditioning is the one done with dogs and with rats, in which animals could integrate information coming from different learning episodes. So he and his team replicated that in an experiment in which animals were given Pavlovian conditioning with one odor. Apple was followed by the presentation of food. This is supposed to endow apple with the properties of an effective conditioned stimulus. But before that, the animals in the group for compound were given presentations of the apple and pear odors. These were presented simultaneously in such a way that the animals could associate them. The control group element or elemental, was given access to apple and pear separately in such a way that they couldn't associate them. In the phase 3, they presented the pear, and they wanted to know whether the pear is well pronounced? Pear. Pear. I struggle with that one. Eh? I should use another fruit. Banana is better. Whether they would show the conditioned response to the pear as dogs and rats do. The results show that the animals acquired the conditioned response to the apple, and when they were tested with the pear, the group given compound presentations of the apple and pear, they showed a very strong conditioned response to the pear, in spite of the fact that this stimulus had never been paired with the food itself. So snails, the ones that you kill in your garden, and you do it, oh my God. They actually are capable of integrating information from different learning episodes. And this is really quite sophisticated. This seems to suggest that snails meet the criteria for unlimited associative learning, and this might suggest that they can be able of experiencing conscious states. Last one, I promise, is with honeybees. Our colleagues that work with honeybees do that by restraining them in a harness, and then they present an odor, which is a conditioned stimulus, and then an unconditioned stimulus, which is sugary water. The animals, in the presence of the sugary water, extend the proboscis, good, and when you pair the odor with the sugary water for a number of trials, when you present the odor by itself, the extension of the proboscis can be considered a conditioned response. So using this procedure, this has been used thousands of times. Some colleagues, Martin Jurfa among them, thank you Gonzalo for sending me the paper, they assessed whether honeybees can actually discriminate a compound from its elements, which requires quite quite a level of sophisticated cognition. So they train the animals in a negative patterning discrimination, where the elements, the other one and the other two, were trained as signals for the unconditioned stimulus, for the sugary water, so the animals extended the proboscis. And then they interspersed trials in which the two others were presented together, but when the two were together, the unconditioned stimulus was omitted. And if the animals learned this complex discrimination, they would respond to A and B, but they will inhibit the response to the compound of A and B, showing that they can separate, distinguish between the compound and the elements. And the result of the experiment is summarized here. The animals responded to A and B, and inhibited the response to the compound. This is quite good. And then to round the result, they also trained the other animals in the positive patterning, where the other one and two were paired with the absence of, were not presented with the unconditioned stimulus, and only when the compound was presented, the unconditioned stimulus was presented. And in that case, of course, the animals responded to the compound, but didn't respond to the elements. And this is brilliant and shows that honeybees can discriminate between a complex pattern and the elements, which is, according to Ginsburg and Jablonka, a key feature of unlimited associative learning. So let me finish. This is my contribution to the field, colors. We tend to agree that vertebrates are capable of unlimited associative learning, and now we can add arthropods and molluscs to this list of species that can be eligible for having conscious states. I would like to add the flatworms, but as I said, I would like to have some more evidence. We've learned that other kingdoms, plants and single cells can actually display associative learning, according to the literature. And there are a number of animal phyla for which we don't have enough evidence. So we don't know really about whether annelids, there is insufficient evidence and there is no convincing evidence that they are showing unlimited associative learning. Nematodes, annelids, echinoderms, rotifera, many of them. Cnidaria and Ctenophora, these are the jellyfish and the like. There is almost no studies about that. I mean, I think that there is one study on Pavlovian conditioning on Cnidaria, but it's old and not really well controlled. So, as a conclusion, there seems to be evidence that different animal phyla can display unlimited associative learning and are therefore eligible to be considered conscious. And this includes the vertebrates, arthropods and molluscs, and perhaps also the flatworms. Many species have not been properly assessed, and in the coming years, and this can take 20 years or 200 years, I don't know, we should be able to produce evidence to complete the picture, at least selecting some species representative of the different animal phyla. We cannot assess all the animal species because there are millions of them, so one per phylum would be more than enough, or better than nothing. This behavioral evidence can be considered alongside other sources of information, neuroanatomical, neurochemical, neurophysiological, whatever you want, to establish whether animals meet the criteria to be considered sentient, to have feelings, or even capable of conscious states. This is the Planaria Lab at Derby. We managed to bring it from Leicester. I was going to say something different. Bring it from Leicester, and this is in a secret location. It's like the Batman cave. In a secret location, you can see here 7 animals being recorded. And the next steps would be to get some funds just to run more of these experiments, writing a review based on this talk, trying to observe second-order conditioning, for example, and this is something that Liam is pursuing right now. And why is this not working? Continue using these animals as models for addiction and anxiety-related disorders. This is something that we've been doing over the last 5, 7, 8 years with Gonzalo Urselay and several PhD students. And the last thing would be to, perhaps, being sentient creatures, we could use the planaria to develop models for pain and pain management, which is one of the great research areas in psychology and the college in this university. And just for the record, last Sunday in The Guardian, they addressed the decision by the RHS, which stands for Royal Horticultural Society. Their decision not to consider gastropods, that is snails and slugs, and weeds as pests anymore. So all the ones that were killing snails in your gardens, shame on you and on me. They have discovered that killing them is more harming than beneficial. They are part of the food chain, and they have discovered, for example, that weeds are essential because the pollinators, the local pollinators, prefer the local weeds over the alien plants that we insist on growing on our gardens. So from now on, stop killing the snails and stop removing the weeds. A bit of something to think about. The research we are doing might contribute to raise awareness about the relatively advanced cognition of animals in general, including invertebrates, the ones that you find in your garden. This might help us better understand what is their place in nature, and if we do that, I'm sure that this will help us understand what is our place in nature. That would contribute to a more respectful and protective approach to our environment. We would be better people, and this would increase our health and our well-being. So don't kill the snails. And finally, if you were thinking, what the heck is all that about? I think that the last couple of slides might suggest ways in which this kind of research can fit within the research strategy of this university. Thank you very much. Thank you.
Thank you very much indeed, Jose, and thank you for the strong environmental message at the end. That a wilder Derbyshire would be better for all of us.
Yep.
And the frogs in my garden love the snails. That's fine. As the university botanist, I never thought today I was going to come and listen to talk about plants, so thank you for that. I'd really like to see those papers.
Yeah, he was pointing at you.
Thanks, Steve. Any questions for Jose?
Can I ask?
Yes.
So I will never look at a snail again in the same way.
Okay.
I'm just thinking, in terms of from an evolutionary biology point of view, if we allow evolution to run its course without any kind of extinction event or whatever, are we saying that all the phyla would make some kind of evolutionary progress towards sentience, unlimited learning, consciousness, and beyond?
Good question. Is the Planet of the Apes question.
It is a Planet of the Apes question.
I tend to think yes. If we don't kill them first, yes. And the common agreement is that life tends to complexity. So, to our knowledge, there is only one biogenesis, and this happened in our planet once, and that's all. There are no several biogenesis, so only once. And this was something very simple, and this tends to complexity. So I've heard many times that when there is life, you can expect complexity to happen. And if there is biogenesis in a different planet from Earth, we can expect it to tend to complexity and generate a civilization. So in principle, I would say that giving it several millions of years, yes, all of them would become sentient, conscious, and bad people like we are. No to the war, by the way. I had to say that at some point, no?
Kath.
This is the Vice-Chancellor of the university, you know.
Hello.
She's a psychologist.
Oh my God. Speak now. Okay. I suppose my question is more about the last statement in how we will use this knowledge. I suppose having spent 7 years where latent inhibition in the Jeffrey Gray lab was everything that everyone did, bar me. And I suppose what I would be wanting to know is where is that taken to us in anxiety through this research? Because I think that does very much fit into the next stages, and particularly pain, because pain's another key area. So just interested, because we do need to look at that, because I think it would be an interesting area for us to develop further, and one that I think we have the beginnings of, and how would we then invest?
Well, I think that given the opportunity and finding the resources.
Well, I didn't know where that.
It's in the Batman cave. I think that it's an interesting thing. I've been discussing it with several people in the community, and there is some interest, and I think that it might fit within the researcher strategies. But whether it will happen or not, I don't know.
I mean, I suppose it can happen if we plan it to happen. And I mean, I spend probably a lot of time now with the trust, and I think, we need to plan it.
Yep.
Because I think it does fit with quite a lot of the other areas. And I suppose I'm interested in how you think that would.
Yeah, but what I would like to avoid, is becoming a collector of learning phenomena, for example. I like to be more strategic and use the knowledge that we have acquired playing with the model to put it at the service of the practitioners. Yeah. So developing models for addiction or anxiety-related disorders or pain management is something that has future. If I continue doing research on latent inhibition and blocking and second-order conditioning, I will retire without glory. Honestly, I think that.
Okay, I don't know if that was a positive or a negative, but I think we probably should look at opportunities. Yeah.
Yep.
Yes.
How is it going, by the way, so far? I'm boiling.
That was a fabulous talk. I really enjoyed it. I'm not a psychologist by any stretch of the imagination, nor a plant biologist. So my question is just a naive question in a way. But with the leaf folding and the response and how it's interpreted as learning, is it simply that the stimuli and the very mechanical presentation on the leaf is causing an increase in receptors onto the leaf, and therefore it's causing overstimulation? And in any sort of system, if you do too much of anything, it'll go to the extreme as opposed to actually learning. And was there any discussion in the papers with respect to that? That's the first question. The second question was, in the experiments with the fan and the light, does the fan affect the carbon dioxide level, or did they actually think about that either? Thank you.
So this is about plants. And I have no idea about plants, to be honest. So
I don't either.
So it beats me. I don't really know how it happens. And what I can tell you as a fun fact or interesting fact is that I didn't show you the whole data that I got. I was interested in knowing whether this change in the behavior of the plant when we were doing habituation is local, or if it affects the plant as a whole, the whole system. And so the last day, fourth day of training, at the end of everything, I asked Jack to stimulate a different pinna of the same plants. And to our surprise, we found that they showed lower level of folding and more rapid reopening than when we first stimulated the target pinna, suggesting that this is a systemic change that we are recording. But honestly, in the absence of controls and preliminary work and continuing with that for a few years, I'm not in a position to say anything sensible. And the second question was about the Gagliano conditioning experiment. The air current was supposed to be a neutral stimulus, and they assessed that, but I don't really know, because the paper is written in such a way that it is really, really convincing, but sometimes I do not get the whole level of detail in the procedures by this lab. So I'm not in a position to really critically assess their procedures. But I think that, I'm very interested in knowing whether other labs would be able to replicate that, and I know that they are looking into it. Probably in a year's time we can have another gathering, and I can tell you about those results. Yep.
Kareem.
I really enjoyed the talk. As an invertebrate biologist, I'm always telling my students what a vertebrate bias research always has. Invertebrates are just one small part of one animal phylum. Hens are just like 20, 30 coming animal phylum. They move on to always bigger animals. So it's fantastic that you've taken a wider. And I think it's a really fundamental question. Where does consciousness begin? And we've had such a human-centric view of the world in the history of research that I think stepping back and actually thinking that we're not actually that different. There's such a strong evolutionary advantage to having the sort of mechanisms that we have to understand the world, to respond to stimuli. Of course, other animals are going to, even plants, are going to actually have the same evolutionary advantage of developing those processes. So I think it's great that you're looking at that fundamental question. I mean, my own son asked me, are humans the only conscious species? Surely other animals are conscious as well. It's such a fundamental question to address. I'm also surprised that there have been so few other studies of classical conditioning in other animal phyla, given how many different animal phyla there are. There seems to be endless possibilities there for redressing that balance.
Yeah, there are many in arthropods, in insects, different types of animals, but there are phyla which have not been even considered. I mean, I mentioned rotifera. I don't even know what rotifera is, to be honest.
I can show you. Thank you. I've got a pond aquarium at home, and I was looking at some with my son.
Okay.
At the weekend, so there could be possibilities there, definitely.
Yeah. Well, I like to hear that you find this interesting and a fundamental question. I can now accept your invitation. I didn't answer to you to give a talk to your students at any point. Sorry about the delay in responding.
Warren.
First of all, fantastic presentation. Really enjoyed it. And as someone who doesn't understand this world, I picked up quite a lot. I'm interested on those 5 conditions for having AL effectively. Does it matter if you enter the world of inorganic materials, manufactured materials that create the same responses? And then does that question then whether those 5 conditions are true or not?
I forgot to I was driving today at some point and thinking I need to mention artificial intelligence. I think that these are fundamental, the 5 or 6 or 10 or 3 key markers for unlimited associative learning are of importance, because then there are systems, artificial systems now, that show learning, that control our lives, and at some point we will have to decide whether they are capable for conscious states or not. And I think that we need to get ourselves some tools to assess the question of whether they are experiencing conscious states at some point.
That's a really common one, but I was also thinking, I was really interested in the work on plants, and before that the work on what was happening within the cell that was exhibiting similar behaviour, and started to think, okay, because my consciousness has always been that you need to network things, and it's the network that creates it. But we have things like shape, memory alloys in engineering that exhibit exactly the same behaviours that you're talking about. You can condition them with 2 stimuli and they respond to one in almost the Pavlovian way. Even at that elemental state, is that a form of consciousness and intelligence that exists in those inorganic materials?
If I could answer your question, I wouldn't be here. I would be in Harvard. Come on. So
it's probably a hypothetical question.
Yeah, no. Question is, if conscience is the product of biological evolution, this would be the product of the biologically evolved conscience producing another system which might develop conscience, but it would be a different sort of conscience. At the end of the day, I'm not sure whether they are going to, there are many film references here. I'm thinking about other movies in which the machines develop their own conscience. 2001 is the obvious one, and take decisions for us, and they might eliminate that. So it's important to keep an eye on that. My son is telling me to...
Well, he's on my message. I think, although there's lots of questions here, I think we have to cut them off. Well, one more, last one.
Thank you very much. You made mention of the potential for pain management in the future research area. Will it be right to say that conditioning and then associative learning is what we use for placebo medicine? And if that is the case, why do we have some placebo experiments failing? Because if I've been conditioned, or if I can associate that like a string needle, probably is coming with a pain medication. Why is it that sometimes when we give some placebo, they fail, if I've already been conditioned? Thank you.
Okay, well, this is a difficult question. Conditioning is part of the mechanisms that produce the placebo effect. And I've been addressing that in some lectures at some point, but I don't know much about it, and I haven't thought about it. But clearly, there is a clear effect of learning, and treatment with a drug and some signals clearly endow the signals as with the properties of an effective conditioned stimulus, and then you can replace the signal. The signal can replace the drug. And how do we articulate that? And how do we put together this with your interests and mine? I don't know, but we can have a chat.
Denise, can I ask you to give the vote of thanks, please?
Thanks, Paul, and a massive thanks to you, Jose, for an incredibly interesting and stimulating, if I may use that term, talk this evening. You've answered questions that I didn't know I'd got, and you have presented it in a way which people, well, certainly myself, have found able to understand, and it's been really interesting and engaging. I will be in the good tradition of income generation selling tickets to the laboratory if anybody would like to join me on a guided tour. And there are a number of questions which I still have remaining, not least of all, how the heck do you get a honeybee into a harness? So, Jose, it sounds as though we could have a complete menagerie in North Tower by the time you've finished. But thank you very much, and I think you've started us on the next part of our journey in learning more about your research and the opportunities that lie ahead. Thank you.
Thank you.